Hello everyone, I'm Miss Priyanka Sanjay Late, working as lecturer at Moa Education Society's Moa Rural Institute of Pharmacy Sone. In this presentation, I am going to discuss topic that is enzymes. This topic we will discuss for first year D Pharmacy students. Content of presentation: Introduction, definition of enzyme, synonym, biological source, description, chemical constituent, collection and preparation, chemical test, and uses of following drugs. First one, diastase. Second one, yeast. Third one, papaya. Now firstly we will discuss introduction. Enzymes are an essential constituent of living cell. They can act independently. They serve a role of catalyzing the biochemical reactions. They are classified according to the types of reaction they catalyze. First one, hydrolysis, for catalysis of hydrolytic reactions. Second one, transferase, for the transfer of chemical group from one molecule to another. Third one, oxidoreductase, catalyze oxidation reduction reactions. Fourth one, lysis, catalyze the addition of groups to double bonds. Fifth one, isomerase, are responsible for intramolecular rearrangements. Sixth one, synthesis, catalyze condensation of two molecules. Many of the enzymes also possess non-protein chemical groups. An enzyme moiety comprises of a protein component, apoenzyme, and a pro-synthetic group representing non-protein component. The latter is also called as cofactor or coenzyme. Certain metals and vitamins also act as coenzymes. The next point, classification of enzyme based on action. In that first one, endoenzymes. An endoenzymes are intracellular enzyme is an enzyme that functions within the cell in which it was produced. For example, synthesis, isomerase. Second one, exoenzymes. Enzymes which are secreted outside cell and act on a particular extracellular substrate is called exoenzyme. Examples, any digestive enzyme like lipase, pepsin, trypsin, etc. Next point, definition of enzymes. Enzymes are the protein substances which serve a role of catalyzing the biochemical reactions. For example, diastase, yeast, papain. In that first one, first good drugs we will discuss diastase. Synonyms, amylose, salivary diastase. Biological source, it is amylolytic enzyme present in saliva found in the digestive tract of the animals, in bracket animal diastase, bracket complete. It is also formed during the germinations of barley grains and known as malt diastase. Several amylolytic enzymes or carbohydrates of commercial importance are known. Many of them are also used in therapeutics. Zymes, maltase, sucrase, cellulase, invertase, hydroronidase, lysoenzymes are amongst the important amylolytic enzymes. Description or general characters. Color yellowish white. Odor faint characteristics. Forms amorphous powder. Solubility soluble in water, forming colloidal solution. pH range 6 to 7. Properties of diastase. Like other enzymes, it is thermolabile and sensitive to a temperature more than 45 degrees Celsius. Diastase is active between 35 to 40 degrees Celsius. It is active in solution having pH range of 6 to 7. Acidic range of pH to 4 causes its deactivation. Diastase catalyzes the hydrolysis of a 1,4-glucosidic linkage of polysaccharides such as starch and glycogen. Diastase can convert 50 times its weight potato starch into sugars. Next point, uses. First one, diastase from various sources is used as digestant. Second one, it is used in the productions of pre-digested stretchy foods and also for the conversion of starch to fermentable sugars in fermentation and brewing industries. Second food drug, yeast. Biological source. It consists of unicellular fungal microorganisms, Saccharomyces cerevisiae, belonging to family Saccharomycetaceae. Description. Color whitish powder. Odor characteristics. Size unicellular microorganism less than 1.5 micron. Manufacture of yeast. Various substrates are used for the commercial production of yeast as mentioned above, but common and industrial use are beet and cane molasses. A suitable strain like S. cerevisiae is grown and it is transferred to sterilized substrate. The equipment is sterilized to get rid bacteria and other microorganisms. The nutrients medium on which the yeast is grown rid of bacteria and other microorganisms. The nutrient medium on which the yeast is grown is continuously added to the culture medium. It is confirmed that the sugar content of nutrient medium is not more than 1%. The pH is kept in between 4 to 4.5 and temperature at 25 to 30 degrees Celsius. Nitrogenous nutrients like ammonium sulfate and sugar phosphate are added to the nutrient medium and fermentation is allowed to take place for about 11 hours. Yeast is separated in centrifuge, washed with sterilized cold water, pressed, filtered and finally dried. The moisture content of the final product is maintained at 75% and it is filled in containers and stored at low temperature. Next point, chemical constituents. The yeast contains about 65 to 85% of moisture from vitamin B group, that is nitrogenous compounds, in bracket proteins, bracket complete, glycogen, fat and vitamins. Vitamins reported are thiamine, riboflavin, nicotinic acid, pantothenic acid, folic acid, biotin, etc. The enzymes invertase, diastase, zymase and maltase are present in yeast. Uses. First one, it is used in manufacture of alcohol, beer and various wines. It is used in bread industry to raise dough. Irritated yeast is used as a source of vitamin D. Fourth one, it is a good source of protein. Third, food drugs, papaya. Biological source, it is cultivated fruiting tree, Carica papaya, belonging to family Caricaceae. Geographical source, it's perennial plant indigenous to tropical America. It is extensively cultivated in Sri Lanka, Tanzania, Hawaii, Florida, and India. Habitat, papaya plant is about 5 meters in height and bears fruits within a year. Fruits are about 10 to 25 centimeters in length, about 5 kg in weight, and dark green in color. When the plant is about 9 months of age, it is used for collecting its latex. Collection and preparation of papain. Vertical cuts are given to the fruits with the help of sharp instruments made of stainless steel or brass. The juice, latex oozing out of the fruits gets coagulated and is collected in suitable containers in bracket plastic, polythene or aluminium bracket complete, dried at low temperature in vacuum. This product is known as crude papain. For the processing of papain, the latex of these fruits is collected in aluminium trays. To the collected latex, potassium metabisulfite in bracket 5 gram per kilograms of latex bracket complete is added. The extraneous matter is cleared out by passing through sieve and the latex is dried in vacuum shelf dryer at 55 to 60 degrees Celsius. It can also be processed by spray drying method. This dried latex is called papain. Next point, description. Papain constituent of papaya. Color, light brown or white color. Odor and taste, typical. pH range 5 to 6.1. Solubility, it is partly soluble in water and soluble in glycerin. Ethyl alcohol and isopropyl alcohol precipitates papain. Isoelectric point of papain is 5.2. It is the most thermostable enzyme known. Next point, chemical constituents. The different proteolytic enzymes present in papaya latex are the mixtures of papain and chemopapain. The proteolytic enzymes acting on polypeptides and amides. Next point, chemical test. First one, 0.5 gram of papaya latex and 10 ml water, shake well, add a solution of potassium permanganate. Decolorization takes place. Second one, 20 ml of milk plus 0.5 gram of papaya latex, curdling takes place. Next point, uses. First one, it is used in classification of beverage and as a meat tendencer. Second one, it is used for degummings of silk fabrics industry and in leather industry for dehairing of skins and hides. Third one, it is used as digestant and anti-inflammatory agent. Next point, storage. It is very sensitive to air, light, moisture and high temperature. It undergoes auto-oxidation very fast and hence should be stored in well-closed and filled containers away from light and in cool place. Thank you.